Hello and welcome everyone to this podcast for genetics. In this podcast, we're going to focus on heredity and some history of heredity. Think about where we've been and how that is leading into our discussions over the next few weeks. Remember, during that first unit of material, we talked about nucleotides. And we talked about how nucleotides could be strung together to make DNA. Also in that first unit of material, we talked about how this DNA could be replicated. Then as we moved on to the second unit of material, we talked about how this DNA could be transcribed, TXN for my abbreviation for transcription, to RNA. And we talked about many different kinds of RNA. And we talked about how this RNA could be turned into a protein through the process of translation. We talked about how going from DNA to protein, it was called the central dogma. We talked about how usually the protein is the trait, or what we might call the phenotype of that organism. But we also mentioned that sometimes the RNA is the final product and is the phenotype that we're looking at. We also talked about how going from DNA to protein, how that could be regulated. In this most recent unit of material, we talked about a few things. We talked about how this DNA here, through the process of meiosis, could be segregated into gametes. We talked about, in this process, how we generated a lot of diversity, genetic diversity, because of the way meiosis works, but we also talked about diversity in the sense of generating mutations on the DNA, and how mutations on the DNA could be incorporated into the RNA and ultimately make a protein that did not fold properly because of the effect of certain mutations. And we talked about, of course, how these mutations can be fixed in the DNA, but we also talked about how certain mutations can lead to cancer. And that's where we closed. In this unit of material, we're going to come back to meiosis. It just keeps coming back. It's the fundamental pathway that we love to hate, but it's so important to understanding diversity, but it's also incredibly important to understanding heredity. How is it that the DNA in one organism is going to be passed on to the next generation? So we'll talk about heredity, inheritance. So in this podcast, we're going to begin our discussion of hereditary and inheritance. And we'll briefly mention Mendel and some other people that were important in setting up the study of heredity. But in the next couple podcasts, we'll talk about Mendelian inheritance, and we'll also talk about non-Mendelian inheritance. Later on in this unit, we'll talk about sex determination, and we'll talk about gene linkage. Okay, so let's go ahead and get started with talking about the history of heredity. So a question that might pop up is, how long have we been studying genetics? And often, when we talk about that, we're really thinking about heredity. But as we've learned throughout the first part of this class, genetics is not just heredity. As a true scientific discipline, it's been less than, say, 120 years. But people have been applying genetics, even if they didn't know they were doing so, 10,000 to 12,000 years ago. In this time frame here, there's evidence that populations, particularly in the Middle East, optimized wheat by doing various crosses. In other populations, other agricultural products, as well as livestock, were optimized by various crosses. If we carry this on and think about other evidence we see in history, over 2,000 years ago, the Talmud, which is the Jewish book of religious laws, they stated that if a mother has two sons who die due to bleeding, then none of her other sons or her sister's sons should be circumcised. Because they knew that if multiple sons were in a family that died because of bleeding after a circumcision, there was some kind of trait that was being inherited that led to this death. So they made a law that future sons should not be circumcised. They also had documented that whatever this trait was, it had to have come from the mother's side. And so that meant her sister's sons were also not to be circumcised. At the time, they may not have known why, but we now know that they were absolutely correct in their assessment, because we know that the hemophilia gene is on the X chromosome. So if she had one mutated copy, that would mean that half of her sons would have the trait and half of her sons would not have the trait. As far as we can go back, we know that there were attempts to try to understand heredity. How do we get certain traits and how do we pass those traits on? In 1665, Robin Hooke, they made the first microscope. It was a simple microscope, but it was a microscope that was sophisticated enough to see cells. So at this point, they could observe cells. With the understanding of cells, scientists were quick to make certain predictions about many of life's processes. But one interesting thing that came up was this idea called preformation. And essentially what this idea states is that inside a sperm or egg, there existed tiny human. There were two groups. There were the spermists, and then there were the ovists. And both groups thought there was a tiny human inside of one of these gametes. So spermists and ovists. The spermists believed that once the mature human in the sperm entered the egg, it found its home, so to speak, and it could grow and continue to develop until it was a baby and was born. The ovists said that the baby, or the miniature human, was in the egg, and once the sperm entered the egg, that energized the miniature human here to grow into what would soon become a baby. Well, we now know that this is wrong. I suspect I did not have to tell you that this was wrong, that you already knew this. But what I want you to do is think about what common observations are there that dispute this idea of preformation. Why is it that we know that the human doesn't just come from the male side or from the female side? All right, the next idea I want to talk about is this hypothesis of pangenesis. And what pangenesis suggests is that if you have an organism, I'll just draw a human because my humans are so magnificently drawn, that within this human there are these things called gemmules. And these gemmules come from all parts of our body. From the tips of our toes, to our hands, to our nose, and they all migrate to our gametes. And once in our gametes, sperm or egg, these gemmules will help determine the next generation, or rather will determine which traits are passed on to the next generation. So if you have brown eyes, gemmules from your eyes will move to your gametes to indicate that brown eyes should be passed on to the next generation. If it helps you remember what pangenesis is, the word pan means all, and the word genesis means origin. So the origin of the next generation comes from parts of all of the previous generation. Though pangenesis is no longer thought to be a correct prediction for heredity, we know that this idea originated in the ancient Greeks and persisted until the late 1800s. Now, pangenesis led to this other idea of inheritance called the inheritance of acquired traits. And from a previous course, you may have heard of this scientist, Lamarck. Lamarck embraced this idea of inheritance of acquired traits. And essentially what the inheritance of acquired traits means is that an organism passes on to the next generation their learned traits or skills. So if someone is a good athlete, the traits that they've learned in that course of that lifetime, their muscle memory, their visual acuity, would gather these gemmules and pass them on to the gonads to make gametes that had those traits in it. If you play a musical instrument very well, those same kind of muscle memory and understanding of being a good musician would be taken from all parts of your body and brought into the gonads. We now don't accept this hypothesis of inheritance of acquired traits. However, as with any good hypothesis, sometimes we come across new data. And this new data is epigenetics. While epigenetics would not explain all of the ideas of inheritance of acquired traits or Lamarckian evolution, it does state that experiences of one individual can alter the genetic expression of one's gametes, which can affect the next generation. We still, by and large, don't accept this in its fullest, but every now and then we see new data that forces us to go back and look at previous data. That's what science is all about. So epigenetics is opening that door, once again, for us to see if our life's experiences somehow can affect our gametes. However, the general idea of pangenesis, where these gemmules come from different parts of our body and end up in our gonads, is now no longer accepted. And that was largely disproven by scientists by the name of August Weismann. So his experiments largely shut down pangenesis. So what Weismann did was he took mice and crossed them. All right, but he didn't just cross mice. What he did was he chopped off their tails, and then he crossed them. Now, if pangenesis was correct, then this next generation would look like this. No tail. And then if he crossed this mouse again with another mouse, again, there would be no tails. But what he found was that this mouse had a tail. If he took this mouse with another mouse from the same litter and chopped off their tails, and he crossed them, again, he would see mice with no tails. Again, he would see mice with tails. He did this for 5 generations, where he chopped off the tails and remated them. And in the end, the only thing he ever saw were mice with tails. If Pangenesis was correct, then at some point you would predict he would start getting mice without tails. Because if Pangenesis is correct, the gemmules from the tail would go to the gonads to direct the development of a tail. He never saw that. We know this today because of other casual observations. If someone loses an eye due to some kind of accident, or loses one of their kidneys, we know that those individuals can still have kids with 2 good eyes and 2 good kidneys. It has nothing to do with the way the organism appears in one generation on how the next generation is going to appear. So if Pangenesis is wrong, then what is the current idea of heredity? And the current idea is the germ plasm theory. So what the germ plasm theory states is that when you have an organism, again, one like my lovely drawn human here, the germ plasm theory says that all of the genetic information is stored in the gametes. Information from different parts of the body are not transmitted to the gonads so that the gametes now have that information. An individual is born with all that genetic information in their gametes. So the germ plasm theory is the widely accepted view of heredity. Okay, it's only taken us almost 3/4 of a semester to get here, but we are finally talking about our good friend Gregor Mendel. So a few things I'm going to write down about Mendel. Now, Mendel is often referred to as the father of genetics. He was born in what is now the Czech Republic. His parents were farmers, so he was raised in a farming family, which perhaps led to his fascination with science and plant breeding. Now, at this time, not everybody went on and finished school or went on to college, but some did. And Gregor Mendel was one of those a few individuals that went on to get an advanced degree. And often what that meant in this time was that they went to the monastery to become a priest. So he went to the monastery and graduated and became a priest. And he did this all in this town called Brno. I won't ever ask you about the town of Brno on the test, but if you're ever there, you may want to go there and visit the monastery, where they still have his gardens that he worked in. After becoming a priest, he went on and obtained advanced studies at the University of Vienna. And at the University of Vienna, he continued his passion with botany and math. He liked a lot of things, but he really liked botany and math. I think it's important to say something quickly about plant science. Sometimes we don't give it as much attention as we should, but it's important to know that many of the early geneticists were actually botanists. And much of what we know about genetics came from the study of plants. And we continue to study plant sciences and genetics to understand how plants work, but also in a more applied way in farming and agricultural sciences. Now, I also included math here because, as we're going to talk later on, we're going to talk about Mendel's crosses. And he certainly wasn't the first one to notice that if you take a round pea and you cross over the round pea, you get round peas, mainly. But he was the first one to combine these two passions of his, botany and math. And because of that, he was able to make predictions about why certain traits were passed on, which turned out to be very important. Now let's return to Mendel's life here for a little bit. After returning from the University of Vienna, he returned to the monastery, where he conducted his famous pea plant crosses. So from 1856 to 1863, he did many studies of pea plants and how to cross them and what you could predict about the crossings. As I look at these dates, I'm reminded of Mendel's contemporaries. That is, I'm reminded of the people that did great things at the same time that Mendel did. So Mendel, Darwin, and Lincoln were all doing great things in this time. They never knew each other. They might have heard of each other, but they never met. And in fact, there's little evidence to suggest that Mendel ever read Darwin's work or Darwin ever read Mendel's work. If they had, who knows what would have happened. But it's interesting to think about what each of these individuals were doing to achieve the greatness that they achieved, all at the same time. Okay, I'll get off my bandwagon there and come back to Mendel's life. Mendel published his work in 1866. He then became an abbot. That's sort of like the leader of the monastery in 1868. And because of all the administrative work he had to do there, he never did science again, at least to the best of our knowledge. And then in 1884, he died. At his death, he was unrecognized for his scientific contribution. And it was not until the early 1900s, around 1900, 1901, that his work resurfaces. And we'll talk more about that later. In finishing our discussion of Mendel, I want to just briefly talk about why they turned out to be such a good model organism for him. Reproduce, they generate lots of progeny. And that's a key trait for all genetic model organisms. They have to produce lots of progenies, and certainly peas do that. Peas are easy and inexpensive to grow. That's also very important. If they're complicated to grow, and it costs a lot of money to grow, it can hinder the investigation. The next one is that peas have a relatively short generation time. It's about 35 days. Now, by modern standards with genetic model organisms, that's not great, but it's not too bad either. Another interesting feature of peas is that not only can you cross them, but you can also self-cross. So by crossing, I mean taking one parent from one plant and crossing with another parent from a different plant. But self-crossing is you can take the same plant, since it has both male and female gametes, you can cross one plant with itself. And finally, peas have easy to visualize characteristics. It's easy to see a round pea versus a wrinkled pea, a yellow pea versus a green pea. So this is very important as well. Okay, on the next podcast, we're going to spend more time talking about Mendel and his crosses. But that's all I have for this podcast. If you have any questions at all, please let me know. If not, I will see you in class.